Welcome back to the Pittcon podcast. In this episode, we speak to Dr. Chad Merkin about next-generation nanomaterials in medical applications.
I'm Chad Merkin, Professor of Chemistry and Director of the International Institute for Nanotechnology at Northwestern University. I've been there for over 30 years, focus on the development of nanomaterials and devices that can advance analytical capabilities, ranging from molecular diagnostic tools to ways of manipulating gene expression to ultimately developing new therapeutic modalities.
Yeah, and how did you first get involved with the International Institute for Nanotechnology?
I started it about 25 years ago. We were kind of on the we're the first International Institute for Nanotechnology, and kind of seized an opportunity as this field was really taking off and began to build an incredible epicenter for this type of research that drew people from all over the world, and that includes fantastic faculty, but also postdocs and students that all contribute to the development of these tools.
Yeah, and what are the main challenges limiting the development of next-generation diagnostic and therapeutic tools?
There are many challenges. It depends on whether we're talking about diagnostics or therapeutics. In the diagnostic front, it's what you can detect, how little you can detect, and where you can detect. And so nanotechnology is so important there because nanostructures give you the ability to build multifunctional materials that can navigate different environments and can bind to targets in unique high affinity ways and can signal in unique ways, all of which lead to new diagnostic capabilities, whether you're trying to do in vitro diagnostics, intracellular diagnostics, or even in vivo diagnostics. On the therapeutic front, similar. It's all about how do you get drug modalities to the point or to the area where they're needed. And that often involves navigating a really complex milieu of tissues and cells and compartments within cells. And the multifunctionality of nanostructures allows you to do that in unique ways.
And what are some of the common areas in the medical field where nanomaterials are currently being utilized?
Diagnostic tools are actually used now everywhere that are based upon nanotechnology. An example is the Merkin system, which was developed by Nanosphere, a company we started about 20 years ago. That was bought by Luminex, which was then bought by Diasorin. That tool is used in over half the world's top hospitals to track genetic markers associated with disease. There are constructs called nanoflares that have been used all over the world to measure intracellular RNA content. And now there are a whole series of tools that are being developed which will become commercial tools that allow you to improve on those capabilities, but also go into uncharted areas, including in vivo imaging.
And are there any nanomaterials that have been particularly prominent in this field, and why is that? An
example of a prominent material is something called a spherical nucleic acid, which we invented a couple of decades ago, 25 years ago. These are constructs that are made by taking little snippets of DNA or RNA, chemically functionalizing them so that they can be put on the surfaces of spherical particles, tiny nanoparticles. And at high density, they're forced to stand upright, and they adopt the shape of the particle core, hence the term spherical nucleic acid. These constructs are the basis for commercial probes used in the Merkin system, for example. They're also the basis for nanoflare technology. They're the basis for fit flare technology. And they're the basis for many new drug modalities that allow you to control gene expression in unique ways, so regulate gene expression, ways to create new vaccines. They can become the adjuvants of vaccines, and when modified with peptides, they can be used to train your immune system to selectively eradicate either infectious disease or to eradicate different forms of cancer. And then they're also the basis for new types of CRISPR gene editing capabilities. So the spherical nucleic acid can enter cells and tissues, unlike normal DNA and RNA, quite freely. And so if you take Cas9, the protein involved in CRISPR or any of the Cas proteins and modify with DNA and load up with the CRISPR components, you now can get CRISPR into cells and tissues that you normally can't get it into, and you can affect gene editing in novel and powerful ways.
That's fascinating. And what roles does toxicological analysis play in enabling nano-based healthcare devices?
Toxicity is always a key concern of anything that certainly is going to be used in vivo, but even things that are used in vitro. You want to know what the basic toxicity of different components are, and being able to figure out and understand where and how you can use these constructs safely is a big part of any exercise developing either probes or therapeutic tools, and they're key in this area as well.
How important is a multidisciplinary approach to workflows that hope to integrate nanotechnologies into the design of a new diagnostic or therapeutic system?
Nanotechnology is the ultimate interdisciplinary field in the sense that it's really about redefining all of science and engineering and aspects of medicine, because when you take a material and shrink it down in size, or if you take molecular components and assemble them up to the nanoscale, those constructs all have new properties that are very different from the bulk. So there's a lot of scientific discovery there, understanding the structure-function relationships. But then there's application. How do I use those properties to develop new tools that can drive biology, that can drive medicine, that can drive clean energy, that can drive optics, electronics, you name it. And so depending on what you're using those materials for, we draw upon different disciplines to really tackle those problems, oftentimes in a very team-oriented way. So it's rare to see somebody really excel in this field that doesn't either have a broad skill set or surround themselves with lots of folks from different fields.
Yeah, definitely. And are there any particular nanotechnologies helping to target devastating diseases like cancer?
There are many nanotechnologies. It's one of the most exciting, I think, areas and advances. Look, if we just went through COVID, those are a step towards nanotechnological tools. So lipid particles that encase mRNA and allow you to effectively trick your cells or teach your cells how to produce antigens that elicit a specific immune response. That's an example of how nanostructures or microstructures in that particular case can play a big role in terms of driving the development of medicine. It gets even better as you begin to build tinier structures and you can control the different components, the presentation of the components, so that you can develop powerful new forms of immunotherapy that fight cancer in unique ways. For example, ways that allow you to not only regulate how these components get into cells, but how they're processed within those cells so that you can maximize the efficiency of a particular type of drug, or in this case, vaccine.
And what are you working on right now that you're particularly excited about?
We're working on vaccines in a big way. So we've been trying to redefine how we think about developing vaccines, not just for infectious disease, but for different forms of cancer, using a concept we call rational vaccinology. And the concept behind rational vaccinology is that structure matters. So if you think of pharmaceutical development over the last, 5 decades, we've focused heavily, in the case of small molecules and biologics, on the structure. Sometimes a single atom can change the efficacy of a drug. With vaccines, we don't do that. It's amazing. We take 2 components, an adjuvant and an antigen, we mix them together, we inject, and we hope for an appropriate immune response. Rational vaccinology says that if I take those components in a nanostructure and present them in different ways, I can get different effects, and I can move along the ineffective to curative axis in a significant way. And so we've shown now with many different animal models that rational vaccinology is real, and that if you take a series of components designed to create a vaccine and structure them in the appropriate manner, you can significantly maximize their efficacy.
And you mentioned COVID before. Obviously, the COVID-19 pandemic has impacted exhibitions such as Pittcon. How important is it for speakers and exhibitors to come together face-to-face at events like this?
Well, face-to-face events like Pittcon are critical. I mean, so much was lost over the COVID years, not just in terms of lab productivity, but in terms of personal contact and exchange of information. Science thrives when there is a lot of interaction between scientists. That interaction is a lot better face-to-face than via Zoom or remote type capabilities.